RNA or ribonucleic acid is a polymeric molecule essential in various biological roles in coding, regulation, and expression of genes. Each nucleotide in RNA contains ribose sugar, with carbon numbered 1 through 5. An important structural component of RNA that distinguishes it from DNA is the presence of hydroxyl group at the 2 position of the ribose sugar. A base is attached to the 1 position. In RNA, cytosine and uracil are pyrimidines. Adenine and guanine are purines. A phosphate group is attached to the 3 position of the 1 ribose and the 5 position of the next. A phosphate groups have a negative charge, making RNA a charged molecule. Synthesis of RNA is usually catalyzed by an enzyme, RNA polymerase, using DNA as a template, a process known as transcription. Initiation of transcription begins with the binding of the enzyme to a promoter sequences in DNA. RNA and DNA are nucleic acids. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, RNA is usually a single-stranded molecule and consists of much shorter chains of nucleotides. The complementary base to adenine in DNA is thymine, whereas in RNA it's uracil, which is an unmethylated form of thymine. Like DNA, most biologically active RNAs, including mRNA, tRNA, rRNA, snRNA, and other non-coding RNAs, contain self-complementary sequences that allows part of the RNA to fold and pair with itself to form double helices. And they are highly structured, but unlike DNA, their structures don't consist of long double helices. The mRNA or messenger RNA is a copy of DNA. Cellular organisms use mRNA to convey genetic information. This directs synthesis of specific proteins. Many viruses encode their genetic information using RNA genome. The coding sequence of mRNA determines the amino acid sequence in the protein that is produced. Every 3 nucleotides or a codon corresponds to 1 amino acid. In eukaryotic cells, once precursor mRNA has been transcribed from DNA, it's processed to mature mRNA. This removes its introns, non-coding sections of precursor mRNA. The mRNA is then exported from the nucleus to cytoplasm, where it's bound to ribosomes and translated into its corresponding proteins form with the help of tRNA. In prokaryotic cells, which don't have nucleus and cytoplasm compartments, mRNA can bind to ribosomes while it's being transcribed from DNA. After a certain amount of time, the message degrades into its component nucleotides with the assistance of ribonucleases. However, many RNAs don't code for protein. Around 97% of transcriptional output is non-protein coding in eukaryotes. These non-coding RNAs can be encoded by their own RNA genes, but can also derive from mRNA introns. Examples of non-coding RNAs are tRNA and rRNA, both of which are involved in the process of translation. There are also non-coding RNAs involved in gene regulation and RNA processing. Certain RNAs are able to catalyze chemical reactions such as cutting and ligating other RNA molecules, and the catalysis of peptide bond formation in the ribosome. They are known as ribozymes. tRNA is a small RNA chain of about 80 nucleotides that transfer a specific amino acid to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. It has sites for amino acid attachment and an anticodon region for codon recognition that binds to a specific sequence on the mRNA chain through hydrogen bonding. rRNA is a catalytic component of the ribosomes. The rRNA is a component of the ribosome that hosts translation. Eukaryotic ribosomes contain 4 different rRNA molecules. In cytoplasm, rRNA and the protein combine to form a nucleoprotein called ribosome. It binds mRNA and carries out protein synthesis. Several ribosomes may be attached to a single mRNA at any time. Nearly all the rRNA found in a typical eukaryotic cell is rRNA. Don't forget to let me know if you have any other question in the comment section below. Bye.